Welcome! In this video, we'll be discussing primer design for your novel transcript variants. How do you know if a transcript variant is going to be novel or not? Well, within the UCSC genome browser, within this viewer, you can compare the reference, either from GeneCode, or also known as the Ensemble database, or from the RefSeq database. These are essentially dictionaries of all the canonically annotated transcript variants. And so you can compare that with your data. In this case, it's long-read sequencing data, but any kind of data that can provide information about potential alternative splicing or potential novel splice junctions will do. So in this case, what we notice in this particular data set, SRR12389274, we notice that there's this novel exon. And the way we know that is by comparing this novel exon here on top with the different exons at the same location in the reference. And we notice that the ending point specifically, so this splice point here, is not shown in any of the transcript variants that are supposedly canonical or supposedly known. So it would be interesting to design a primer pair, or to design a primer pair that spans this long splice junction between these two exons. Because if we could amplify the specific sequence in between these 2 points, we would be able to verify that this is in fact a novel transcript variant that is expressed within this particular cell. The particular cell type is from 293 T cells. How do we design the primer? Well, a primer, as a recap, is a short piece of DNA that allows you to bind to a specific portion of a sequence, and then you can utilize a polymerase in order to make a copy, a complementary copy that goes all the way to, as long as the polymerase will go, or as long as the insert sequence is. The insert sequence being the sequence between the forward primer and the reverse primer. And then the way polymerase chain reaction works is that you make many, many copies of the insert sequence, with the primers basically serving as the edges of those insert sequences. Great. So this is going to be our forward primer on this novel exon. Let's see how to make a primer for that. In order to zoom in and look more on a base pair level resolution, we can utilize the Shift key and then drag with the left click over the novel exon. This will prompt us to zoom in and look at that particular region, which we will do. And we can see here up top that there's a sequence. Now this isn't exactly very user-friendly to work with. There's no way to copy it from here. Also, we see that the scale is 10 bases. Primers are typically between 18 and 24 bases. So we might not have a lot of flexibility here with what we're looking at. So we're going to zoom out a little bit over the exon. Now we have maybe a 40 to 50 base pair stretch to work with, and we can pick 18 to 24 bases anywhere along this stretch in order to have a primer that binds to this novel exon. Okay, so if we want to get the DNA in a little bit more of a user-friendly form, we go up here to the View tab, and we view the DNA. It will prompt us some information, but this is actually already all automatically entered. All we have to do is click Get DNA, and then we have all of the information that we need in front of us. And so here we see a lot of useful information. It seems like over here there's a large stretch of A's. Those are generally repeats with lots of T's around. If there are large repeat regions, you generally want to avoid that in a primer, because oftentimes this will then bind to many different places that have long stretches of A's. which is almost every RNA, or long stretches of T's. So we also want to keep in mind all of the other requirements. So we'll check these, more systematically in a little bit. But one that you should be thinking of as you're picking maybe the first 18 to 24 bases that you might want to try and think about as a primer is that you want it to end with 1 to 2 GC base pairs. So as we mentioned in the lecture, GC, the interaction between guanine and cytosine, is stronger than the interaction between adenine and thymine. And so you want to make sure that the primer binds strongly near the end of that sequence, because that's right where the polymerase is going to start acting. And so it's very important that you have a tight interaction there. So perhaps this GG here would be a good place to put a primer. Now I'm going to pick what looks like 18 to 24 base pairs. I'm not sure. And then I'm going to check all of these other requirements. In order to do that, we utilize a sequence manipulation suite, or basically a website called bioinformatics.org, where we can just paste in the sequence, submit it, and then get information about it. And so what we learn here is that the sequence that we randomly picked is 24 bases. It's got a basic melting point of 61 degrees, and it passes all of the other different components. So the GC content is 58.33%. That's all good. So why is that good? Well, we have the requirements here. We need to be between 40 and 60% GC content. The PCR primer suite said we were at 58%. The length should be between 18 and 24. Okay, so in this case, it's 24. It's a little large. The transcript source, well, that's just mentioning the fact, or the locus that we're looking at, in this case the link 173 locus. It starts and ends with 1 to 2 GC base pairs. The melting point, well, it's not between 52 and 57, it's 61. So maybe we need to go back here to the drawing board and cut out a base pair from the start. See how that changes it. So now there was this G that was a part of it. It was 24 base pairs long. Now we've removed the first G, and we've got only 23 bases. The sequence manipulation suite, well, we got it down to 59 as our TM. Not quite perfect yet. Let's remove, just in here, another base pair from the start. See what happens. And we see that we made it down to 57. Okay. But we have a T as the starting base pair. It would be better to have a G or a C. So we'll remove one more base, and then we've got a basic TM of 56 that fits all of the other statistics that we're looking for. GC content. There's the GC clamp, so the fact that there's 2 C or G's at the end, and everything else. So great, so we'll take this as our primer. Since this is the forward primer, it should, the forward primer basically refers to the fact, when we look at this large base pair, that the forward primer binds onto the reverse complementary sequence. So this sequence here binds to the sequence on the reverse strand. The DNA is double-stranded. We're looking at one strand within the genome browser, and we can bind to the other. And so we can utilize the sequence that's just given to us here in order to bind to that other sequence, which is not shown in the genome browser. So we have our primer sequence. We've checked all of these various components, and we know that they're all the way they're supposed to be. And we can also say that this is a forward primer, because it's the starting point of our insert. And we'll just give it a random name here, test 1. You'll want to make the test name something that you can write down easily on a test tube. Alright, so we've got the primer sequence, and we have this column called specific. in order to know that the primer sequence is specific, we need to make sure that it doesn't bind to any other genomic regions or loci. So in order to do that, we utilize a tool called the Basic Local Alignment Search Tool, or BLAST for short. And all that you really need to do is perform a nucleotide search here, enter the accession number, and you'll want to enter in the organism Homo sapiens. Because we're working with human cells, so it's just important that it doesn't bind to anything really within humans. All of the other settings you can just leave as is and hit BLAST. Usually this might take between 30 to 60 seconds. It's searching through a lot of different sequences, a lot of different information. So we'll wait. And now we've got here the results, which indicate that, in fact, the number one hit is link 173 and related sequences. That's good. And then if we look specifically at what alignments are otherwise working, we see that there are some unspecific transcript variants, but only for 16 out of the 21 base pairs. So this is a little bit of a conundrum. Is this particular primer specific? It's specific for 6 out of the 22 base pairs, but not for all 22. But we can say that on the basis of the query cover being 76%, the e-value 5.6, we can probably try this primer out, see whether it works in order to amplify what we're looking to amplify. And if you get unspecific amplification, well, you can point back to this as perhaps being a reason why, and you'll have wet lab evidence for once you run the experiment, you'll have wet lab evidence for whether the unspecific binding matched the prediction here. So there isn't a correct choice in this instance as to whether to redesign the primer entirely or to work with it. This is kind of a gray zone, but for the purposes of this video, we'll say that the primer is specific enough. So specific enough. And then instructor verified means that your instructor, principal investigator, whatnot, has verified that the sequence you designed matches. Typically, they'll do that also by examining it in a BLAST search or using a tool called BLAT, and then figuring it out. Okay, let's design a reverse primer. So looking back at our large sequence, we designed a forward primer here, and the forward primer is pointed in to the right. We're going to amplify an insert that contains this large splice site. So we have sequence on the other exon that is over here to the right. You can see here, right, that this space is approximately 1 kilobase. And so when you look at this sequence, there's certainly no way that it's 1 kilobase. The whole sequence, in fact, is only about 450 base pairs long. But here, all the way at the back, maybe the sequence or the insert we can roughly approximate as 200 base pairs. Eventually, we're going to calculate this, but for now, you should try and design inserts that roughly exist between 200 to 1,000 base pairs. Those will be the easiest to amplify with PCR. Generally, the shorter the sequence, the easier it is to amplify. The longer, the more complex, the more optimization that needs to go into the experiment. as in the number of tries that you need to do with the PCR goes up. So in order to design the reverse primer, we'll pick some region here at the back so that the insert is about 200 base pairs. We'll zoom in. We've got this sequence. The scale is 10 base pairs again. We might want a little bit more. So to zoom out, now the scale is 20 base pairs. So there's about 40 base pairs here to work with. We view the DNA as before. We get the DNA. And now we have this sequence to work with. And then we can pick, as we did before, approximately 18 to 24 base pairs. See whether in our first attempt we're anywhere near that. We are. We picked 20 base pairs. And we have here the basic TM as 50. We want it between 52 and 57. So the basic TM is a little low, but all of the other things are fine. Okay, so let's try that again. If we add a few more base pairs in front, we can see what happens. We submit this. We see we're at 52. All of these different things still pass, but it looks like we're going to need at least a few more base pairs in order to get something or a TM in the range that we're looking for. So we submit a few more, and now we see there's other warnings because we have a lot of self-annealing and hairpin annealing. So okay, the basic TM has gone up to 52. That works, but we have this annealing. At 23, we've reached all of the different components that we're looking for from a technical standpoint, but then again, this primer may not be the best primer since, it's a little bit longer, has a lot of A and T in it, whatnot. But we've reached everything, except the GC content. The GC content is not between 40 and 60%. Generally, for GC content, if it's below 40%, that's a little bit better than if it's above 60%, because once it's above 60%, you will get a lot of unspecific binding because of these strong interactions sort of dominating the behavior of the primer. But it's very close to 40%. So this primer may be appropriate. However, since it's a reverse primer, we can't just use this sequence. As I mentioned, the forward primer back over here points to the right. And we need a primer that will point to the left in essence, right? A primer that will bind to the sequence that is shown in the genome browser. And so in order for it to bind to the sequence that is shown, and by the sequence that is shown, I mean when we, zoomed in, we saw the sequence up here. We need the reverse complement of that. We need the part that is going to bind here on the other side. So in order to reverse complement, we put the raw sequence into here. We submit it, and now we have the reverse complement results. We can put this into our PCR primer stats again. It won't have any particular effect, right? Because A goes with T, C goes with G, so all of the bonding, all of the binding patterns, that's all conserved. But now we have the sequence that we should actually post in as our reverse primer. And now this primer will point to the left. So we have one primer that points to the left, one primer that points to the right, and we can amplify what's in between those. So we'll name this primer test 2. The source is link 173. We hit all of the different components that we were interested in hitting. And then, of course, we say that this is the reverse primer because we reverse complemented it. And now the question is it specific? Well, we copy this sequence as we did before into the nucleotide blast. We have the Homo sapiens as our organism again. We hit blast. We wait. And then we see that, like the last one, right, we have another thing that it matches to, but only for 69% of the bases. In terms of here, the sequence of interest, link 173 and its related sequences, we get basically 100% match. So the percent identity for here, 16 again. So like the other one, we have this little complication, a little bit of a gray area as to whether to use the primer, but since it's only 69% and the E value is greater than 1, we'll say that the primer matches and is suitable for our experiment. If it isn't, we'll figure it out. We'll learn that at another point in time. But for now, we'll say that that is satisfactory. So specific enough. Great. So now we've designed a forward primer, a reverse primer. As we design primers, we can order them and then perform our wet lab experiments. Thank you very much.